Okay, Dr. Massani, I think you're okay now.
Yeah, I think we have a lot of participants, which is great. Okay. So welcome to the today's weekly research conference. Very pleased today to introduce Dr. Peter Liu on his talk entitled Novel Link Between Immunity and Mitochondria, Key Regulators and Heart Remodeling. As before I would like to introduce him with a speaker's bio, but I have a few housekeeping notes before to tell you before we begin. So number one, you have to submit your questions through the Q&A, not the chat. They'll be answered at the end of the presentation. We'll also be accepting live questions at the end of Dr. Liu's presentation. So just use the hand up feature and you will be asked to unmute by the moderator, myself, and use the chat feature if you are experiencing any difficulties. So, and I want to remind you, there is no research rounds next week due to the American Heart Association meeting. So no research round next week. So, of course, it's a great pleasure and honor to introduce Peter. As you know, Peter is our Chief Scientific Officer at the Heart Institute. He's also Vice President of Research here at the Institute. Peter has a tremendously successful career, and he was the inaugural director of Heart and Stroke Richard Lewer Center of Excellence at the University of Toronto. So we could steal him from Toronto. And he also was, after his role in Toronto, he was director of the Institute of Circulatory and Respiratory Health at the CIHR. He has launched a lot of networks, imaging, clinical trials, knowledge translation. And actually, Peter is very well established worldwide and international level through these networks. He's very, very well known. So he has been, of course, the architect of the Oracle Plan, the Ottawa Region Cardiovascular Research Strategic Plan, which leads our research and is a framework of all what we do in research at the Heart Institute. And we know we came from different stages of implementation. He has also, of course, a large scale programs. He has many awards, including the Research Achievement Award and Lifetime Achievement Award at the Canadian Cardiovascular Society, the Rick Gallup Award from the Heart and Stroke Foundation, and Institute of Circulatory and Respiratory Health Distinguished Lecture Award from CIHR, and the Margulies Award from UBC, amongst, of course, many others. He's well known for his contribution to heart failure, cardiac inflammation research. He has discovered basically how viruses can enter the myocardium and trigger inflammation, and how innate and acquired immunity contribute to cardiac remodeling and heart failure progression following heart injury. Most recently, Peter has received the CIHR Rapid COVID grant to conduct a randomized controlled trial, which was great news for the Institute. He has actually already published several papers in high impact journals on the impact, such as circulation, on the impact of COVID-19 on the heart, and his expert opinion in this area has been sought by very prestigious scientific journals, as well actually by highly respected mainstream media as The Guardian. So I could spend a long time describing achievements of Peter Liu. I think most of you know him very well, but it's really a great pleasure to ask him to do this wonderful presentation that everybody's very excited about. So thanks, Peter.
Yeah, merci beaucoup, Thierry, and thanks so much for the kind words. And thank you all for joining us. And I'd like to share my screen if possible. And just to, is this the right framework? I just want to make sure that you're seeing some fall colors here, and this is the full frame.
Yes, it looks good.
Okay, great. Yeah, thank you so much. And certainly this is a special time of the year, and we're traversing through many moments in history as well. And but I'd like to share with you some recent data that was just published in Circulation in how we think about inflammation that may actually link with the metabolism in the heart and its relevance in the setting of heart failure. And I thought this will be interesting to share with the esteemed audience and get some feedback in terms of how we might use this information going forward. So I really appreciate the kind support, and give a shout out to Dr. Hanbin Lin, who's a postdoc in our lab, who has done a lot of the recent work in this area. And of course, this is a special week as well. And this is a week of the Remembrance Day. And we certainly know that many sacrifices, especially during the wars, are very important legacy for our country. And the poem, In Flanders Field, is well known by many Canadians, and authored by Colonel John McCrae. But not everybody may know that, in fact, he was a physician, and in fact, looked after many of the wounded during the war. And this is, I think, a particularly fitting tribute. But of course, there are many other losses, including COVID-19, as Terry was mentioning earlier. But we must not forget that there are other ongoing challenges in health beyond COVID-19. And one of the major examples is heart failure, which is the major chronic cardiovascular disease that we all face. And indeed, more than a decade ago, when we actually first look at the Canadian data, we appreciated the fact that, in fact, heart failure comes in different forms. The more traditional form is the heart failure with reduced ejection fraction, typically after an episode of myocardial infarction leading to cardiac remodeling, enlargement, and with fall in ejection fraction and consequent heart failure. But we called attention to the fact that there is actually rising alternate form of heart failure, which is heart failure with preserved ejection fraction, often coming after hypertension, diabetes. And in this situation, one can actually get hypertrophy, fibrosis without necessary dilatation, and thus the ejection fraction does not fall, but yet the outcome is just as dire. And unfortunately, more than a decade later, we still face the major challenges in heart failure, and that 1 in 5 Canadians face a lifetime risk of succumbing to heart failure. We have now over at least 1 million patients living with heart failure. 1 year mortality, unfortunately, still 25%, even though we now actually have more evidence-based therapy, certainly for heart failure with reduced ejection fraction. And we certainly know also that this is a major burden in terms of hospitalization. 30-day readmission rate in Canada still 21%, and is commonest cause of unexpected hospitalization. And that the patient on the average stay 8 to 10 days in hospital with just in-hospital costs of over 2.8 billion per year. So this is a real major challenge. There are still many forms of heart failure that actually has no treatments. So the question is, are there other windows of opportunity that we can explore to address this? So today I'd like to share with you in terms of the pathways that can lead to cardiac remodeling, and maybe some novel factors that can actually lead to remodeling that actually plays fairly major role, and a complete novel link that has both the innate immunity and the metabolic regulation in one single setting. So what we always are very aware of is the fact that the heart failure is not a one-step process, in that there are many factors, risk factors such as the traditional cardiovascular risk factors, genetic factors, and also ischemia and other injurious stimuli to the heart that can actually lead to the remodeling process, which is really host response to these risk factors, whether it's the hypertrophy process secondary to pressure load, whether it's injury from ischemia or from toxins that leads to immune activation and subsequent fibrosis. And then it's really actually the end stage that comes to our emergency department when the patient's no longer able to compensate in terms of cardiac function, that the patient presents in terms of shortness of breath, pulmonary edema, peripheral swelling, etc. And so this is really actually a long journey, and the opportunity truly resides not necessarily just when the patient presents to hospital, which is really at the end of the long journey, but in fact, understanding the processes that actually led up to heart failure, in which we may be able to detect this process better with markers or imaging, and so that we can institute treatment and reverse the condition earlier. And in this nice review by Professor Sadashima's group, in which we can actually see that with these type of injury, multiple processes takes place. But keeping in mind that the heart can actually favorably remodel, for example, in the setting of exercise, elite athletes, in which the vasculature keeps up with the change in the cardiac shape and the function, and that the heart, in fact, is very well preserved and function hypernormal in that type of setting. But it is really actually in the adverse setting in which there is actually buildup of contractile elements, whether in parallel, for example, in the setting of hypertrophy, or in series, for example, in valvular regurgitation, in which the heart dilates up, that leads to the adverse remodeling with the inadequate blood supply, with increase in inflammation that ultimately leads to the adverse consequences. What are some of these processes that we are now getting more familiar with? So indeed, we do know that there are a host constellation of processes that actually is perturbed, that is in existence in the failing heart. We know that there are processes such as cell growth. There's also process of cell death. There's increased fibrosis, abnormal calcium regulation, as well as metabolic regulation. And some of these, when understood, can indeed become targets for therapy. For example, in terms of cell growth, we do know that the agents such as the renin-angiotensin modulators, as the ACE inhibitors or sacubitril valsartan, can actually modify the hypertrophy process and mediate good outcomes in the right patient. In addition, we do know that the contractile abnormalities are critical in terms of the setting of heart failure. But however, addressing this pathological process is very important in terms of modality. We know that giving inotropic agents that increases calcium is actually harmful, but there are potentially alternative strategies, such as the agents that increases active myosin interaction, such as omecamtiv mecarbil. And the large galactic trial, heart failure trial, will be presented this weekend at the American Heart Association meeting. And so that this is really an exciting progress in this domain. But on the other hand, we also know that the cell death, reactive oxygen free radicals, fibrosis, are really consequences of inflammation. And the questions, are there ways in which we can actually address this very important process? And certainly studies with the IL-1 beta blockers such as canakinumab give us a very kind of exciting, tantalizing suggestion that this may be in fact modifiable and important. But also a very important aspect is the metabolic dysregulation, which is very common in patients with heart failure, very well described, and certainly team here at the Heart Institute have documented with PET imaging the abnormalities. And the question is, are there ways to modify this? Maybe the SGLT2 inhibitors, but this is not clear at the moment. So we can see that there are major processes at the present time that's in play in the setting of heart failure, yet we really actually don't have much sort of a handle in terms of how they are regulated. To address some of these, we have been using system biology tools such as proteomics, trying to understand through the type of biomarkers that's actually released in the setting of heart failure. And for example, this can be done either using mass spectrometry or protein chips of different varieties, in which the larger amount of data can be then organized, analyzed using statistical approaches or the machine learning AI approaches to organize into pathways, which then can give us clues as to the best biomarkers that may be able to allow us to not only understand the disease, but also to devise treatment. And some of the details were recently published in the European Heart Journal. And indeed, when we actually look at some of these biomarker candidates, whether in this study, which came from the Swedish group or from our group or from Dr. Mann's group, that you can see that there are a large number of biomarkers that are very important in terms of looking at the outcome. When you actually look at the quadrants, the increasing propensity of predicting outcome versus the patient's status in terms of New York Heart Association. Some of the best markers indeed are part of the inflammatory pathway. But currently, we don't actually quite understand exactly why inflammatory markers are released in the setting of heart failure, even without infection, for example, or the fact that there are such good prognostic markers, and what's the regulation that actually allow these to be activated. And looking at a lot of the system biology data together make us to actually revisit our original concept of the classic heart failure reduced ejection fraction or heart failure with preserved ejection fraction, that it is not actually a dichotomous, sort of a black and white process or red and blue in here. But in fact, this is a dynamic process because we do know that in fact, patient does not necessarily have to stay within those categories. Because if we can actually understand the etiology that leads to the patient's heart failure, whether that's due to ischemia, due to valvular disease, due to underlying rhythm abnormalities, or given the right treatment, for example, using beta blockade or sacubitril valsartan, one can actually change the ejection fraction in the patient and actually begin to unpack some of the processes that actually led to patient's compromise. And so, but on the other hand, if these processes are unaddressed, then patient will actually continue to progress. So this is where we think that not only by understanding the clinical factors, but understanding the biological factors through, for example, appropriate biomarker combinations, that one can actually identify the underlying pathways, whether it's inflammation, fibrosis, metabolic abnormalities, cell death, that allow us to then reach the opportunity to address these with precision treatments to allow us to then change the natural history for that patient with much more thoughtfulness rather than just giving everybody every agent there is available. And in particular, we'll concentrate on the inflammation and metabolic consequences today. Now, sorry about this busy slide, but this is actually the state of the art understanding of the pathways that actually leads to cardiac remodeling. And really congratulate the authors in terms of putting this huge effort together. But we can actually quite readily recognize, for example, major pathways such as the renin-angiotensin system through the angiotensin receptor that goes to the calcineurin NFAT pathway, including MAV2, that leads to the hypertrophy process that we know so well, and that the agents that can actually block this receptor or block the production of angiotensin 2 are effective in the setting of heart failure. In addition, there is a complementary pathway involving the catecholamines, particularly the beta adrenergic receptor, which is through adenyl cyclase can activate the cyclic AMP pathway, or through the MAP kinase pathway can also piggyback onto cardiac hypertrophy. And certainly we are well aware that the beta blockade, for example, is very effective in terms of attenuating this growth process and some of the toxic consequences. In addition, there are mechanical sensors such as the TRP channels, which also utilize the calcineurin or CAMP kinase pathways, and that leads to increase in hypertrophic response, and of course, reducing pressure, for example, in the setting of aortic stenosis can be very helpful. There are some counter-regulatory pathways as well, such as the natriuretic peptides, and certainly Heart Institute here has done a lot of work in terms of natriuretic peptide, and natriuretic peptides taken advantage of, for example, through sacubitril valsartan, or cyclic GMP, which is taken advantage of by the recent agent, the vericiguat, can also help to attenuate some of these processes. But you can see that there is actually a large segment here involving metabolism and the inflammation, but it is actually not really connected to the rest of the pathway diagrams, and the exact activation of these processes and also how they are linked together in terms of upstream signals are not really quite as well known, even though we do know that they are important players, certainly in the progression of heart failure. So this is part of our journey and the quest to understand better in terms of where the inflammation signals come from and how the metabolism, in fact, may be linked into that. Some of the background to this come from our earlier work in terms of understanding how viruses interact with the heart. And this is one of the review articles, and with co-authors Slava Eppelman and also Doug Mann, in which we have mapped, for example, in terms of virus going through directly its receptor. This is a Coxsackie virus, but very importantly is that the virus interact with the intracellular receptors such as MDA5 or RIG-I, that these then can link to the activation of NF-kappa B, which is the master transcription factor that leads to downstream inflammatory response. But in addition, the viruses can also engage the innate immune receptors, whether that's the toll-like receptors 2 and 4, for example, that's on the cell surface, or could be actually in the endosome, particularly toll receptors 7 to 9, and that can then converge onto signal transducers such as IRAK4, then that also can lead to NF-kappa B activation and the inflammatory response. But there are also other pathways which are counter-regulators and which are important to consider, and particularly the endosomal toll receptors can actually activate the interferon regulatory factors that leads to interferon production, which is, of course, these are important players nowadays when we are trying to understand the SARS-CoV-2 or the COVID-19 virus and how they interact with the cell systems, and including the heart, that can lead to long-term consequences. But but this system is just as important in the setting, for example, of myocardial infarction, in the setting of hypertension, diabetes, and in terms of recognizing a lot of stress pathways, but without necessarily, for example, a pathogen like a virus. And we do know that these intracellular pattern recognition receptors are becoming more and more important because they tend to recognize not just the viruses, or bacteria, but in fact, the altered proteins or DNA that's inside these cells through long-term consequences of these risk factors. And in particular, a very important novel set of intracellular receptors are the NOT1 RIP2, which interact very closely with the RIG-I, and which can very importantly activate and after signal amplification. Indeed, NOT1 and the NOT2 are important intracellular signal for innate immunity recognized through mutations that are found in patients, for example, with various inflammatory conditions, particularly inflammatory bowel disease. And so it turns out that in these settings, this happened to be intestinal epithelium, and in which we know that there are a lot of bacteria, and certainly the microbiota is very, very important in this type of setting. But there is a delicate balance in terms of the pathogens or endogenous microbiome with the intestinal cells. And this is in fact monitored through two systems. One is the NOT1, NOT2, RIP2 system, which constantly monitors the level of these pathogens, and then be able to actually activate through signal cascade in terms of inflammatory response, for example, through NF-kappa B related activation. Similarly, the inflammasome system through our P3 and ASK also can lead to appropriate inflammatory response, and to check the microbial pathogens in imbalance, so that keeping in mind that if there is excessive activation of these systems, you get the inflammatory bowel disease. But on the other hand, you have an inadequate response, you also get a bacterial invasion, which can also be detrimental. So this is a very important regulatory system. And it turns out that the NOT1 and 2 are these important intracellular pattern recognition. It's very important to recognize these yellow colored components of these moieties, and that's the card domain. And that's essential for all these intracellular innate immune signal transducers, and which stands for caspase recognition domain. And these are very important later on when we talk about how we discover MAVS in this process. So on the other hand, we also know that in the setting of chronic heart disease, whether that's due to ischemia, due to pressure overload, there are a lot of so-called danger associated signals or altered, for example, DNA, RNA, or proteins, which then can actually act as stimulating antigens with the right receptor, can activate the inflammatory response. But what is not known, certainly even at the time of review, is whether some of these novel pattern recognition receptors, such as NOD1 and RIP2, do play a role in this type of a setting with chronic injury or stress to the cardiovascular system. But keeping in mind that NOD1 and RIP2 are well positioned to potentially carry out this signal amplification and response process. So initially, Koichi Naito and also then later on Hanbin Lin from our lab have actually taken up the task of trying to understand whether NOD1 RIP2 in fact can be an important player in the hypertrophic remodeling leading to heart failure. And this is a situation in which actually take a mouse heart in which we create pressure overload by constricting the aorta with in a very controlled manner, increased pressure in the left ventricle, and therefore leading to, in the appropriate setting, with left ventricular hypertrophy and subsequent heart failure. But then we try to understand the role of NOD1 or RIP2 by taking knockout animals in which NOD1 and RIP2 are genetically deleted to see if that makes a difference. So just a general survey of in terms of what this model looks like. Certainly that if you look at the heart in the wild type, the heart after banding is actually very significantly enlarged, and cross-section shows a very significant hypertrophy. There's a decrease in ventricular function. There is also increase in heart cell size representing the hypertrophy. There's also increase in inflammation and also fibrosis as indicated by the pink color using picoserious red representing collagen deposition. Now, what happens if we actually genetically remove NOD1? Does it make a difference? We can see that the hearts are smaller. There is actually less hypertrophy. The myocyte size is smaller. The fibrosis is less as well. And the interesting RIP2 knockout, keeping in mind that the two of them tend to work as a partner, also shows almost identical type of phenotype. There's a protection, and therefore suggests that NOD1 and RIP2 may be in fact contributing to the significant degree of left ventricular hypertrophy and subsequent heart failure we see. So for example, if you actually look at the measurement of heart weight to tibial length ratio, you can see that in the knockout animals, there's a definite reduction in terms of hypertrophy, suggesting that these signals are contributing to the hypertrophy process. Very importantly, we also look at lung weight to tibial length, which means that looking at the lung water, which is a reflection of the amount of failure, typically like pulmonary edema. And we can see that in these animals with removal of NOD1 RIP2, there was actually almost complete protection in that there's no accumulation of lung water in the animals, suggesting that there is protection from heart failure. And indeed, when we actually look at the other components, they are very, very consistent in terms of protection, in terms of removal. Now, the question is, this may be just an animal model phenomena. Does that happen in patients? And indeed, we do know that the NOD1 RIP2 downstream factors are upregulated in patients with heart failure. This is part of the reason we went to look for this. But indeed, when we actually look at human hearts, for example, the explants or biopsy, who has heart failure, that the NOD1 and RIP2 are also increased. So this suggests that this is actually very relevant in the setting of human heart failure as well, in addition to the fact that with the serial banding, you get increased expression over time. What is most impressive to us was the fact that when we see that after banding, if you actually look at survival, there's definitely increased mortality in severe banded hearts because of heart failure. But those animals that had NOD1 or RIP2 removed, you can see that there was actually significant protection with about 50% reduction in mortality. And this is a big deal because very few treatments can have this such a beneficial effect. And this is also replicated by looking at the cardiac function, looking at fractional shortening, for example, significant decrease in the wild type animals. But if you actually have NOD1 RIP2 removed, there is improvement in terms of fractional shortening, in terms of heart size, very much dilated in the wild type. There is actually reduction in ventricular size, or even look at biomarkers such as BNP, which is the best prognostic marker for heart failure. And there was a significant reduction in terms of BNP levels in the heart for these animals. So really, there is actually very broad, consistent protection when you actually have the ability to actually attenuate NOD1, RIP2, which are the intracellular innate immune activators. And so apologize for this fairly busy slide, but it's really just to show that when you actually remove NOD1 or RIP2, you get actually decrease in terms of inflammatory signal activation, whether that's involved components of NF-kappa B, or if you look at TRAP6, which is a very important downstream signal for inflammatory activation, that in the wild type, there's significant upregulation, but in the knockout of NOD1, for example, whether at 2 weeks or at the 8 weeks, there was very significant attenuation, suggesting that the inflammatory activation is very much reduced. But similarly, in terms of hypertrophy signal, for example, for phospho-ERK1 and 2, there's actually also a decrease in the knockout animals. And this is fairly preserved as a pattern, for example, in the RIP2 animals as well, really suggesting that the NOD1 and RIP2 have almost duplicative type of effect in terms of phenotype, and really attesting to the fact that the two of them work together as partners. So if we actually look at just a conclusion as what we have so far, it suggests that NOD1 and RIP2 are upregulated in the stress heart, contributes to mortality and progression of heart failure, and that NOD1 RIP2 deletion attenuated the hypertrophy signal transduction, as well as NF-kappa B related upregulation in terms of inflammatory response. And therefore, it suggests that this is an important regulatory unit in the setting of heart failure. But what was interesting is that we were trying to figure out, in fact, where NOD1 RIP2 are located in the cells, and trying to figure out who are the other, partners in crime, so to speak, in this type of setting. And if we had to guess, we'll say that most of this probably is going to be around the nucleus or around the ER, because that's where NF-kappa B end up going. And so you can see that it's interesting that when you actually do these myocyte tracking experiment, if you take the myocyte and expose them to phenylephrine, which is the most precise hypertrophy stimulator in this type of setting, what we have seen using confocal microscope is that the NOD1 and RIP2 begin to actually co-localize, which does not happen actually when you don't give the hypertrophy signal. So it really suggests that this is very specific to the hypertrophy stimulus they get together. But what was interesting is that they did not converge around the nucleus as quite often we see, but in fact, they converged around mitochondria. And for example, we use a tracker such as a mitotracker or other labels of mitochondria that in fact NOD1 RIP2 get together, but then sit around in mitochondria. So this is a bit of a surprise, why would these innate immune signals sit around the mitochondria? And this is where a bit of a detective work that Hanbin and others have done in the lab to figure out what could the partner on the mitochondria be to actually attract the NOD1 RIP2 complex. And indeed, they went through many informatics searches, big data, and then look particularly at some of the structural components. And one of the key clues was the fact that both NOD1 RIP2 have the card domain, as we mentioned earlier. And so they were also looking for card domain containing entities around the mitochondria. It turns out that the top candidate and the most dominant card-containing domain moiety on the surface of the mitochondria is this protein called the mitochondrial antiviral signaling protein, MAVS, which I didn't even know that they have anything to do with hypertrophy, but nevertheless, this is the key card domain containing innate immune protein on the mitochondria. And so we try to learn something about MAVS and to see how it's got anything to do with hypertrophy. Well, certainly there's no data to suggest that it has anything to do with hypertrophy in the literature, but MAVS has been recognized before, indeed, as an important antiviral protein on top of mitochondria. And it has the capacity to interact with, for example, a viral entity, and through RIG-I, which we talked about earlier as an established pattern recognition receptor inside the cells. And it can also interact with many downstream TNF, for example, signal processing intermediates that ultimately leads to NF-κB activation. It can also interact with TBK1, for example, and then in turn lead to interferon regulatory factor activation, such as IR7, IR3, lead to interferon production, and can also interact, for example, with STING, which is a separate story that we're working on at the present time. So you can see that indeed MAVS is very important in terms of coordinating the cellular response in terms of an antiviral or antibacterial insult. But exactly how MAVS interact with the rest of the immune system, how they actually interact with the mitochondria actually has not been much explored. And indeed, people who work in MAVS area recognize that there are other partners that needs to be identified in order to have the complete story. And I should also mention that, in fact, it's very important that a regulator sit on top of the mitochondria, because the mitochondria under stress conditions can undergo either mitophagy, in which there is a significant amount of damage, and the removal of the mitochondria through the lysosomes, or mitochondrial biogenesis, in which the mitochondria can actually link together and regenerate a healthy mitochondria. And this balance is actually critical. And we'll talk about momentarily what MAVS does in this type of setting. So does this antiviral protein have anything to do with heart failure? So we went back also to our heart tissue of human hearts that suffered heart failure and look at MAVS. Well, lo and behold, in fact, MAVS does increase actually in the setting of human heart failure. And then not only that, when we actually look at mouse heart tissue after banding, yes, MAVS is also upregulated, just like what we've seen before with NOD1, RIP2. So MAVS indeed tracks with the severity of heart failure. And even though there's absolutely no infection here, no virus, no the traditional trigger for MAVS activation, even though it's an antiviral protein. And so then we also look at the reprogrammed human cardiomyocytes, thanks to Dr. Wenbin Liang as their expert team. And so you can see that these myocytes exposed to phenylephrine, which is a hypertrophy signal, they will enlarge, and the NOD1, RIP2 are increased, and MAVS does actually increase along with other hypertrophy signals that you would expect. And the other thing that was interesting that if you then take these cardiomyocytes, expose them to phenylephrine, and if you knock down MAVS, for example, in this case, using an siRNA, many of the hypertrophy signals, in fact, gets decreased. So it suggests that MAVS was not just a pure receptor of stress, but in fact could be a mediator of hypertrophy in this setting. And so what then it appears that MAVS in fact be the counterpart on the mitochondria of what NOD1 RIP2 would be in the cytoplasm as a danger signal receptor, and that they can then help each other to amplify when there is a danger signal, and so it's in the cytoplasm. NOD1 RIP2 will be activated if they actually involve mitochondria. Then MAVS can be activated. And so you can see here, if you're actually looking at confocal, look at NOD1, RIP2, and the MAVS, the NOD1 in green, RIP2 in yellow, and MAVS in red. And you can see that under normal circumstances, when there is actually no stimulation, such as hypertrophy with the phenylephrine, they don't have anything to do with each other. They mind their own business. But on the other hand, when there is phenylephrine, when there is a hypertrophy, well, then they begin to get together. And you can see that there is co-localization of NOT1 and RIP2, and there is also co-localization of NOT1 with MAVS and RIP2 with MAVS as well. So you really suggest that this is actually at least a tripartite complex involving all these players, and that they interact with each other, possibly through the CAR domain. And this is where the regulatory process begin to actually take place. So what could be the MAVS function when it's activated versus if you actually remove MAVS impact on the mitochondria, because it probably has something to do with the mitochondria if it's sitting on the mitochondria. Originally, we were thinking that, well, if there's inflammation, maybe MAVS will increase mitochondrial output. You know, you need more energy when you're sick and things like that. But it turns out we're completely wrong. And so, for example, when you actually look at the sRNA with phenylephrine, look at complex 1 and the complex 3, and you can see that in this setting with phenylephrine, there is some activities of complex 1 and 3. When you actually remove MAVS, not adding MAVS, but remove MAVS with the sRNA with phenylephrine, you get actually marked increase in complex 1 and complex 3 levels in this setting. So in fact, MAVS appear to put a damper on the mitochondrial action. Is this true? You know, if you look at the complex 1 activity, for example, MAVS here is in purple, MAVS knockout, knockdown or knockout in purple color. And you can see that in terms of complex activity, let's say this is calibrated at 100% in the resting state in the control setting, and that when you add in the NOT1 knockout or RIP2 knockout, there's actually increase in complex 1 activity. And in fact, the maximum amount of activity takes place when you knock out MAVS. So it appears that these inflammatory signals tend to dampen mitochondria. And does this actually have impact on energy production, such as ATP? Well, it's exactly the same pattern. And you can see that under control conditions, if this is kind of the standard, and you can see that with MAVS knockout, there is actually very significant increase in ATP production. So indeed, this system of activation appears to actually dampen mitochondrial function. Of course, this is what we see in setting of heart failure. You know, we get increase in inflammation, but then we also get marked reduction in energy output, and that's why the heart is failing. Even though intuitively we think, you know, the heart should be, doing the opposite. And in addition, as I mentioned earlier, it's actually very significant damage to the heart that you can actually get a fragmentation of the mitochondria, and then the mitochondria can undergo mitophagy, which then can be actually reprocessed, and the new mitochondria can then be generated in this type of setting. And indeed, we were interested in knowing, for example, does the NOT1 RIP2, particularly MAVS, contribute to the mitophagy and mitochondrial fragmentation? So this is in the control setting in which there is no activity in terms of mitophagy. This is a mitophagy dye, which indicates the fragmentation of mitochondria together with lysosome. And when you find mitochondria fragments in the lysosome, that's a reasonable indication of mitophagy. And indeed, when you actually add, for example, phenylephrine hypertrophy signal, you can see that there is actually mitophagy taking place, and then indeed the mitochondria signals are inside the lysosome, suggesting that they're being gobbled up by the lysosome. But on the other hand, if you actually give sRNA, knock down the MAVS so that the MAVS on the mitochondrial surface are not necessarily so activated, even in the presence of phenylephrine, there's actually no evidence of mitophagy or minimal evidence of mitophagy, suggesting that in fact, in this type of setting of hypertrophy, downregulation of MAVS is in fact potentially beneficial. And many of the players that is very important in terms of mitochondrial biogenesis, whether the PINK1 or mitofusin 1, mitofusin 2, are also downregulated, certainly in terms of banding, in terms of hypertrophy. So suggesting that this is consistent. And also in terms of, for example, cell death signals that are coming from mitochondria, such as BNIPs, 3, which is generally increase in the setting of banding and hypertrophy stimulus. This is completely attenuated when you actually remove MAVS in the MAVS knockout animals. So it really actually suggests that there is an inappropriate response in terms of mitochondria in the setting of inflammatory activation following stress, and that the removal of MAVS actually has a very significant protective effect. Does this happen in the setting of human situation? Thanks very much to Han Kim, who actually looked up the large GTEx database involving over 300 humans in terms of RNA-seq data. Oh, sorry. And one can see that in this setting, because we're interested in knowing in this type of setting, if you actually have increased the MAVS expression, what are some of the other partners that goes up? And indeed, we can see that in the MAVS activation, you get ATG5, suggesting autophagy is increased when you have MAVS activation, BNIP3, which is cell death, and BECN1 is also autophagy, and PINK1, metafusin 1 or 2, suggesting that all these processes we are observing in the myocytes and in the animal models are also activated in synchrony in the human hearts. So suggesting that this is a pretty consistent biological process. Maybe I'll just kind of in view of time, just to go to the bottom line here. And so then the other aspect the lab did, and thanks to Liang Zhang and David Smythe together with Hanbin, in which we try to ask the question, is it the CARD domain that's actually the critical signal regulator in this type of setting? So we took wild type NOT1, removed also the CARD domain, as well as RIP2 in terms of removal of CARD domain, and look at in terms of pull down experiments. And then you can see that I think the most important experiment here is that when you have NOT1 here versus the CARD domain deficient version here, that you can actually see that the wild type pulls down MAVS. So it really suggests that NOT1 directly interacts with MAVS. But if you actually remove the CARD domain, well, MAVS doesn't come down anymore. Similarly with RIP2. So it really suggests that CARD domain is the critical component that actually bring all these parties together. And it's also consistent the function of the CARD domain. Does this actually have any impact on the metabolism, metabolic sort of efficiency in this type of setting? And thanks to Ben Rothstein's team, in which we used a small animal PET machine to actually measure the glucose metabolism in these hearts following banding. So just the orient here, blue represents low glucose activity, and red represent high glucose activity. Keeping in mind, in setting a heart failure, glucose is the preferred substrate in this type of setting. And you can see that in the wild type animals, there is a relatively low glucose utilization at baseline. Following banding, there's some increase, but it's really actually not very, very dramatic. But on the other hand, if you actually remove NOT1 or remove RIP2, and in these hearts, you can see that there is a marked increase in terms of glucose uptake. And so this really suggests that these hearts are much more metabolically efficient, and that they are able to therefore potentially produce more ATP as a result, and therefore being able to deal with the stress and the pressure overload in this case, and to protect against heart failure. So finally, then in conclusion, that we can see that through this series of experiments, that the NOT1 RIP2 signaling is actually quite essential to cardiac hypertrophy and failure progression. So keep in mind, these are intracellular pattern recognition activators. And so these are intracellular innate immune signal partners. And that they can then recruit MAVS on the mitochondria, and then in turn regulate NF-κB and the hypertrophy signals. So that this really actually is a very critical complex that coordinates danger detection, and also in terms of the pathway regulation downstream. And the MAVS activation actually inhibits mitochondrial energy production and increase mitochondrial fragmentation. So this process, in fact, is detrimental, certainly for the heart, from the heart failure point of view, and that this complex represents a novel regulatory link between innate immune activation and mitochondrial energy balance under stress conditions. So this is relatively new. We're quite excited by this because we now, in fact, may find the kind of the critical key that unlocks the connection amongst all these systems, which has been a bit kind of a mysterious so far. And the inhibition of this pathway may be actually a very attractive therapeutic target for heart failure, because you can actually improve energy production, restore mitochondrial health, and be able to, in fact, reduce a lot of these inflammatory processes that leads to fibrosis. So very much appreciate the amazing work from the lab and also collaborators. As I mentioned earlier, Hanbin Lin, our postdoc, and the earlier Japanese postdoc Kotaro Naito, who's now back in Yokohama in Japan, has done the lion's share of this work together with lab members, Leon, David, Chujiang, Guohua, Mo, Gori, and Alice. And we are particularly thankful to our collaborators, University of Ottawa, Dr. Mary Ellen Harper, who of course is the expert on mitochondria, Hank Kim for the GTEx work, Ben Rothstein for the beautiful PET work, Wenbin Liang for the reprogrammed human myocytes, and their team members allow us to really put all the story together. And also Dana Philpott at the University of Toronto, a long-term collaborator and really the world expert on NOD and RIP2 as well. So with that, I'd like to conclude and really appreciate your attention and be happy to entertain any questions.
Thank you, Peter. All right, so does everyone hear me?
Yes. Yes, no problems.
Okay, I'm just, okay. I have actually, so congratulations, Peter. That was a really fascinating talk and just amazing work. I mean, I think it's very hard to kind of imagine the amount of work and that is put together by your group and all your collaborators and through your network. So we have actually a lot of questions here. So I'm going to start with one question from Jiaohui Jia. The question is, does heart failure also lead to increased glycolysis in addition to decrease of mitochondrial respiration? Acute inflammation such as LPS response switch the ATP production of glycolysis.
Yes,
Peter,
yes. Oh, absolutely. Yeah, so that's a very good observation. Indeed, in the setting of heart failure, you know, obviously, originally the heart's preferred substrate is actually fatty acid and acetic acid and you know, those components. But in fact, in the setting of heart failure, it tends to be more glucose oriented. But glucose oxidation is still the dominant energy source, and we see that switch here. But glycolysis can also be increase as well. But of course, glycolysis is fairly inefficient in terms of energy production. So the amount of ATP produced is quite low, and the glucose oxidation is better. But of course, you know, if we can restore a Krebs cycle, will be the best.
Oh, excellent. So I have a question from Katie Reiner, who first congratulates you. Great talk, Peter. Very interesting. I'm wondering what you think of the differences between ATP mitocomplex function in the MAVS knockout versus the RP2 lockout you showed. It suggests they aren't entirely in the same pathway in the mitose. Any ideas?
Yes, absolutely. So we think these are really partners in crime. And in the setting of stress, so under resting conditions, when everything's good, everybody do their own business and nobody minds, you know, sort of bother anybody. But under a situation of stress, certainly with pressure overload with phenylephrine, and these are classic cardiac stresses, it brings them together, in fact, on top of mitochondria. So that's why they tend to share the same function, but they do actually very unique things. For example, NOD1 regulates calcium as well, but the other players don't do any of that. And the RIP2 obviously, you know, have other functions in terms of immune and the cell survival and death function, but then the other players don't actually do that. MAVS seem to actually concentrate on the mitochondrial regulation, but they, under stress conditions, they work together. So they have many of the shared characteristics, but on the other hand, each one has a unique contribution to this whole dynamic process. Yeah, so we learned a lot in this program, a lot more to learn. Yeah, so it'd be great to get your input as well.
Okay, so we have a question from Sylvain Boet. So thank you, Dr. Liu, for sharing amazing research. I'm curious to hear more about the role of reactive oxygen species as one of the possible links between inflammation, mitochondria, and remodeling.
Oh, absolutely. So in fact, that's how we started. Hanbin was very interested in terms of, reactive oxygen species. How does that actually regulate, the remodeling process? And of course, the largest source comes from mitochondria. And that's the reason that, all this end up in the mitochondria. So Hanbin is happy in the end. But what happens is that the reactive oxygen species, depending on where they are located, have different function. So obviously, when they are actually activating death domains, they can actually lead to cell death. If they actually are on structures, they can actually modify, whether that's the lipid bilayer or the proteins. But in the mitochondria, they can actually interfere directly with the mitochondrial function. So what happens here is that we now begin to actually find out what are the specific regulators are in these type of setting. And as you know, the problem with free radicals is if you just give antioxidants, nothing seems to work. So you really actually need to target the source of these. And this is why we now, want to land on the switch on the mitochondria.
All right, so thank you, Peter. We have another, they keep coming. So how is MAVS related to not only Mr. Tandy, how is MAVS related to mitochondria energy production? And how is the NOD1 RIP2 MAVS reflecting the energy role of mitochondria?
Yeah, so the MAVS effect on the mitochondria was the completely opposite of what we thought. Originally we thought that when you actually have this system activated, there's the inflammation, you need to therefore drive up the mitochondria, which actually happens in a setting, for example, with the infection. We think what happens with this system, that this is actually a counter-regulatory system. When there is a bacteria, for example, the mitochondria is overproducing free radicals trying to kill the bacteria. But unfortunately, this can actually kill the cell. So what happens is that when you have immune activation, the MAVS is coming in to actually toning down the mitochondria so that it doesn't actually destroy the entire cell, and so that you have more focus, the ability to get rid of the pathogen. So that's great, in the viral infection situation, in the bacterial infection situation, but it's not so great in the setting of heart failure. This was a process in which you tone down mitochondria and, but then unfortunately in the failing heart, it actually needs more energy. So, what nature has designed for one purpose turns out to be not quite suitable, for this purpose. And so this is why we think that, retuning this response may be actually helpful.
And we have Ramana Vaka, who is asking actually 3 questions. Thank you for the great talk. Number 1, we go one by one, okay? Is there any link between MAVS and NLRP3 inflammasome, since NLRP3 inflammasomes is known to be activated at the mitochondrial membrane?
Yes, so we haven't explored fully that component yet. There is a definite crosstalk between the NOD system, that's actually quite well known, and because they actually, structurally share similar motif, and you can see that in many cells, they actually share a parallel function, but then of course the target site is different. One actually lands on ultimately in terms of NF-kappa beta, but also in terms of the mitochondria, whereas IL-1 beta, can become, the cytokine pathways. But they are definitely coordinated. And so we are now trying to actually figure out exactly where the crosstalk takes place. That's a very, very good question, yeah.
And second is, have you looked at mitochondrial ROS in this setting?
Yes, sorry, we didn't show that data, and Hanbin actually very specifically look at that, and it's exactly as you would expect. in that. So the interesting thing is that rather than is when you actually inhibit MAVS, rather than actually a lot more ROS taking place, in fact, it's actually relatively attenuated. So this suggests that in fact MAVS is a purposeful regulator in terms of mitochondrial function, rather than just kind of releasing the mitochondria to produce endless amount of ROS together with the energy production. Yeah, so that's why we think this could be a very good therapeutic target.
And the last question on this one is, how do you think the NOD2 RIP are increasing complex 1 and 3 function, like in terms of mechanism of action?
Yeah, so we're not quite sure exactly what the regulation is, but it seemed to be that these inflammatory signals, so that comes all the way from NOD1 in terms of RIP2 MAVS, as you can see earlier, that they actually regulate each other's production. So when you bring down one, the others come down as well. So there is actually very close coordination of how they function. But at the same time, they seem to also regulate the mitochondrial component as well. We mentioned earlier in terms of they appear to regulate mitophagy and the mitochondrial biogenesis. I didn't show the data, but in fact, MAVS is very important in terms of how mitochondria link with each other. And you saw earlier, the impact on DRP1 and the mitofusin and things like that. Yeah, so so it appears that these complexes, in fact, quite deliberately regulate the components within the mitochondria in addition to the function that we have seen.
I think we have back Joey Jia with the last question. What's the underlying reason for mitochondria changes their behavior, such as the RIP2 recruitment to MAVS due to heart failure?
Yeah, so I think we kind of developing this concept in that the system gets activated as part of response to stress. And I think the nature has designed this system, I think, to be protective so that it doesn't allow mitochondria to overrun. So it actually puts a damper on the mitochondria. And it also tends to process the mitochondria through mitophagy. So it's all kind of a carefully regulated kind of stress response. It's a bit like a cleanup engineer in a way. But what happens is that in the setting of heart failure, you actually need a lot well-functioning mitochondria. So this system, is kind of a little bit of too much of a kind of environmental control. Sorry, I shouldn't say that at the moment. And so in the setting of heart failure, you need to actually let loose a little bit so that you can actually preserve the mitochondrial function rather than, just turning them down. And so I think this is the paradox that we're actually realizing. Of course, there's a lot more to learn in this whole process.
So thank you, Peter. So I just have actually from my own side, more like a clinical type of question. So I think this is fascinating research. And how do you envision the translation to clinical practice? I guess it would be in several steps, right? You would imagine first having a biomarkers, kind of early diagnosis of the disease, so to try to tackle the clinical, before clinical symptoms happen. And lessons of COVID have, for instance, is that when you start to see symptoms, it's probably too late. And it's better to if you tested patients and positive that they have no symptoms, and you would be able to detect some kind of, with biomarking, things happening to further down the road, you will save a lot of lives and prevent probably many things to happen.
Yeah, absolutely. That's perfect, perfectly put, because we know at the present time, the reason heart failure has so much high mortality, despite, you look at the clinical trials, yes, great, we're making an impact, but, you look at the control arm, even the treat arm still has so much mortality because we're treating these patients way, way too late. And so this is why, but the nice thing is that, these inflammatory markers are well elevated. So, but right now we don't do anything with that information because we don't have, much understanding even why they are up. And those, we just kind of don't pay attention to it. But in fact, the information is right there.
But we could imagine, let's say, if you have an early detection because the only treatment that has proved to be reasonably successful actually during this COVID crisis was dexamethasone. So we could imagine that we could do early treatment with dexamethasone before anything else, and it would actually cure the disease. Anyway, at least prevent people to die from it or to be very sick with a lot of sequela. And because we don't know yet, people who had COVID and within 8 months, 12 months after COVID will be alive, but with a lot of problems with their lungs and potentially their myocardium. Anyway, I think we could talk for hours about that. It's fascinating research again. Thank you, Peter, for your great work as a scientist and your leadership and for research. I thank you all for attending the meeting. And I guess for those who have more questions to Peter, it's not very hard to get in touch with. So send him an email. I'm sure he'll have a lot of good answers for you. Thank you, everybody. Thanks, Peter, again. Thank you. Have a great week.